Hello, my name is Tamra Mendelson, and I'm an evolutionary biologist at the University of Maryland, Baltimore County. My team and I study darters, which are small freshwater fish that are native to eastern North America. Darters are found near the bottom of streams, and they dart around while looking for food or when they get scared. These fish are colorful, and during the breeding season, the males become even more colorful. Their colors become bolder, more saturated in order to attract females. Generally speaking, I'm interested in the processes that make nature beautiful and diverse. And I'm interested in how all animals, not just humans, perceive beauty. So how does that translate into scientific research? My research focuses on understanding how darter visual perception affects their decision-making, and especially how they choose their mates, and how evolutionary changes in color patterns and preferences for colors lead to the formation of new species. So for example, if one population chooses to mate with the greenest individuals, while another population chooses to mate with the most orange individuals, these populations will be unlikely to interbreed. And over many generations of isolation, individuals in each population become genetically distinct from one another. And at some point, they become distinct enough for us to consider them different species. And in that way, the evolution of eyes and the cognitive brain systems for interpreting visual stimuli are directly connected to the evolution of mate choice decisions, which then leads to changes in biodiversity, like how many species of fish there are, or how much variation in color or patterns or shape or size or behavior there might be within these species. In your activity today, you'll be analyzing actual data from a study my lab published in 2013 that was led by Dr. Tory Williams as part of her PhD research. And here's Tory to tell you about it.
Hi there. A primary goal of this project was to test whether a female darter's visual sensitivity to certain wavelengths of light predicts their mating preferences. But first, let's back up. Humans have three types of vision cells called cones in our retina that allow us to perceive different colors of light. These three cone types respond to either short, medium, or long wavelengths of light, abbreviated as S, M, and L. And then the resulting curve, called a sensitivity curve, describes how much a cone gets activated or excited by a particular wavelength of light. And our brains then interpret the combination of cone excitations as different colors.
So if the light reflected by an object and hitting our eyes is mostly in the short wavelength range, that activates the S cones, and our brain sees this as blue. If the object is reflecting medium wavelength colors, that activates M cones, and our brain says, oh, that's green. Long wavelength colors activate L cones, which our brain interprets as red. But in nature, everything's variable. I mean, have you ever argued with someone about the color of an object? Each of us sees the world in slightly different ways because of differences in the way we sense and process light. Some folks have higher sensitivities to certain colors than others. And on the other end of the spectrum, some folks, about 1 out of every 12 males, has some form of colorblindness that makes it hard to distinguish between two colors, usually red and green. Colorblindness is most commonly because a genetic mutation in the M cones causes the sensitivity curve of the M cones to overlap with the sensitivity curve of the L cones. So that makes these two images look the same, for example.
People with color blindness have three types of cones as well, but due to an inheritable mutation, one type has a shifted sensitivity curve. But in the darters that Tamra and I study, there are only two kinds of cones.
Darters don't have the short wavelength sensitive cones. So they are most sensitive to shades ranging from green to red, with little sensitivity in blue and violet colors. For our study, we were curious about how sensitivity to different wavelengths of color affects mate preference. Our hypothesis was that females prefer the colors that they're most sensitive to. So specifically, we thought that females would prefer a model fish that is painted a color that most strongly stimulates the cones in the female's eyes. For
the experiment, we made a plastic model shaped like a darter And we painted one of the models orange, which means that the light it reflected had an average wavelength of 570 nanometers. And then we painted a different model, a reddish color, and it had a reflectance of 584 nanometers. If our hypothesis was true that females prefer colors that their eyes are more sensitive to, they should spend more time swimming near the model that excites both the medium and long wavelength cones.
Peak sensitivity in this case would be the place where the M and the L curves overlap, so about 550 nanometers. The orange model's average reflectance at 570 nanometers is closest to this, so if our hypothesis was correct, females should prefer the orange model.
To do the experiment, we captured wild darters by netting them in the Barren River in Monroe County, Kentucky, and Line Creek in Clay County, Tennessee. We brought them back into the lab and set them up in gravel-filled tanks that mimicked their native habitat. For each experimental trial, we placed a live female in a 37-liter tank with a smaller tank on each side of the large tank. And each of those smaller tanks contained one of the model fish, one red, one orange. The models were attached to servo motors that made them appear to swim, mimicking a live male. The larger tank with the female had markings dividing it into 3 parts: a red and orange preference zone closest to the red or orange models respectively, and a neutral zone in the middle indicating no preference. An observer used a computer program called JWatcher to keep track of how long the test fish spent in each of the preference zones over a period of 20 minutes. We then could calculate the proportion of time that each female spent in the red preference zone versus the orange preference zone.
Was our hypothesis correct? The orange model should have been more stimulating than the red model. Did females in fact prefer the orange one? Analyze our data and find out for yourself.